For recordings of neural and muscular field potentials, electronic recordings from a pair of bath electrodes are synchronized with high-speed video recordings and displayed on a laptop monitor. A juvenile crayfish is placed in a small aquarium and allowed to acclimatize for 5 minutes. The animal is touched with a handheld probe by the experimenter after the recordings have been started, and the resulting behavioral and physiological responses are measured and subsequently analyzed.
Hi, I'm Dr. Jens Herberholz from the Department of Psychology at the University of Maryland, College Park. Today, we will show you a procedure to record non-invasively neural and muscular field potentials in freely behaving animals. We use this procedure in our laboratory to identify neural circuits that are activated during escape responses in juvenile crayfish. So let's get started.
To begin constructing the recording electrodes, 0.5 to 1 mm of insulation is stripped off one end of an insulated copper wire. This process is repeated on a second identical wire to create a pair of recording electrodes. Using non-toxic glue, the recording electrodes are attached to the inside of a thin-walled glass recording chamber. The recording electrodes are positioned opposite each other in the center of both short sides of the chamber. Next, the ground wire is made by stripping 2 to 3 centimeters of insulation off one end and soldering this end to a longer piece of wire.